Sandra Kaufmann is a cell biologist who for the last 7 years has been the Chief of Pediatric Anesthesia at DiMaggio's Children's Hospital. She is also the founder of the Kaufmann Antiaging Institute and the author of the book, The Kaufmann Protocol, Why We Age and How to Stop It. In the book, she describes the mechanisms of aging on a cellular level and the best supplements to slow down its progress. And with that, let me start the interview. So hello, Dr. Kaufmann. Thank you for joining us today and welcome to Modern Healthspan.
Thank you. It's my honor to be here. Thank you for having me.
So you have a protocol that you designed kind of from the ground up for retaining youth, I think could be how we describe it. And we'll definitely want to get into that. What I'd like to do is to talk a little bit about your background and what led you to develop this protocol in the first place.
Oh, absolutely. So I will tell you that it was the most selfish thing that anyone could ever do for themselves. I was hanging off of a cliff one day because I'm a rock climber, and that seems to be things that I do a lot. And it dawned on me in my mid-40s that unless I solved the problem of aging, at least for myself, my days of hanging off of cliffs successfully were probably doomed. It takes a bit of strength and youth to do these things. And I love them so much, I didn't want it to stop. So I turned to my background, which I was a cell biologist. I'm a physician now. So I deal with how the body works, how cells work, how drugs affect the body, and how the body affects drugs sort of on a daily basis. And I set out to sort of unravel why we age. And it was probably a little bit nuts when I started doing it. And my office kind of looked like a Post-it note factory had exploded. There was just stuff like everywhere. It was kind of nuts. But I decided that your cells age in 7 specific categories, and that if you took it apart, you could decelerate the process in the individual categories, and that by summing it up, therefore, you could decelerate all of aging. And I started doing this, of course, on myself, and it seemed to work. And all of my lab studies got better. Everything about me got better. And then people around me in the hospital started asking what they could do, and it sort of begot itself. And then once I realized or decided that you age in 7 specific categories, I would look at all of the agents that people sort of put out there as being longevity substances. And I wanted to know exactly what they did in each of the categories. So I ended up with this big, giant piece of paper, because I'm not of the computer generation, with pluses and minuses. Like, what does agent X do in category 1, 2, 3, 4? If it worked, it was a plus. If it didn't, it was a negative. And then it really worked, so it became 2 pluses. And all of that turned into a rating system. So now every agent that I analyze comes with a 7-digit Kaufmann rating number, and it tells you exactly how efficacious it is in each of the 7 categories, right? Which then means if you take your list of things that you're taking and you make a grid and you add up all the numbers at the bottom, it'll tell you how well you're doing fighting each aging category. So basically, it becomes more of an algorithm than an actual science.
Right. So you, just going back a little bit, so you said that it worked on yourself. So can I ask, what did you use to track that? I mean, what kind of markers?
So as physicians, we talk about subjective and objective markers, right? Subjectively, I'm suffering from the same things that everyone suffers from as you get older. You're more tired, you have less energy, your muscle tone starts to go down, your vision gets a little bit worse, your hair gets a little bit worse, that sort of thing, right? Aches and pains after a hard workout, and all of that disappeared. So that's my subjective. Objectively, I measured everything that was sort of stereotypically easy to measure, lipids, basic metabolites. I didn't do the more complex things like the sirtuins and the NAD levels, because at the time it wasn't available. But everything that I could measure, I did, and everything seemed to get better.
Right. Would you have like specific ones that you think are most important in the blood marker kind of area?
I think it depends on who you are. I think that people that have very high incidences of cardiovascular disease, your lipid panel is going to be more important. If people are more worried about hormonal failure, menopause, testosterone loss, all that sort of thing, then that sort of becomes more important. The farther I've gotten into this, the more esoteric I think the labs have gotten. And I think right now your interleukins are more important and your sirtuin levels are more important because it really tells you what your cells are doing, not what the outcome of the cells are doing. Does that make sense?
Yes, no, that makes sense.
Because in one of my categories, I talk about the fact that the sirtuin pathways, the genes and then the proteins that come thereafter, get shut down as we age. And we know that there's 7 of them. And we know that if you pump them back up, things go better. But, you know, if you go to your standard lab section right now, no one's going to measure your sirtuins unless you come to a longevity place and then you can. So the labs have just become more specific over time. And since I didn't have them when I started, I can't tell you they've gotten better because I don't have a baseline.
Right. Okay. So you alluded to these 7 tenets that you use to kind of think about aging. So could we go through what they are, and how did you actually? So how did you decide on those 7 particularly at the beginning? What was kind of the process?
So what I did, I didn't really know where to start because I didn't know where I was going when this whole thing started. This was just me being a very curious cell biologist. And I pulled pretty much, I don't know, thousands of cellular articles as to why cells don't do well over time. You know, various people have come up with other ways of looking at it. They look at proteostasis and this and that, the other. But I decided, as I went through all of my Post-its and then sort of slid them into various categories, that some things overlapped and some things didn't. And it was more like a big Sudoku puzzle. But I'll tell you what I ended up with. And it's probably easier to see how I got there by looking at the end, right? Okay. So tenet 1, I call DNA alterations. So all the things that can go wrong with your DNA over time. And classically, this is going to be telomere shortening and then epigenetic modification. And then recently, I've added the idea of DNA protection, right? So some people would put those in other categories, but I thought, you know, what goes wrong with your DNA? Clump 1, DNA, right? That's the information of your cell, must be protected at all costs. That's super important. That's bucket 1. So bucket 2 is mitochondrial failure, right? You have to have energy for your cell to work. And a lot of diseases sort of are processed around the idea that you don't have enough energy. So why do mitochondrial fail over time? Well, so they fail because of shortages of things. So the biggest thing is nicotinamide deficiency. And most people over 40 are nicotinamide deficient, right? And there are a few other deficiencies. There's coenzyme Q deficiency and a variety of other ones. And then secondly, in this category, it all has to do with processing of oxygen. So when you develop free radicals destroy things. Your mitochondria make endogenous substances to get rid of free radicals, but over the course of time, you don't make enough. So you have a failure of catalase and superoxide mutase and all of those. So the idea is if you can bolster your own endogenous free radical scavenging systems, you're going to do better overall, right? So in the mitochondrial category, those are the big 2. Of course, there are other reasons that your mitochondrial fail, but that's basically tenet 2. Tenet 3 is something that I call pathways. And there are pathways genetic to protein issues that fail over time. And I talk about the big 3. The first one is your sirtuins, the 7 mammalian sirtuins. The second is the AMP kinase pathway. And then thirdly is the mTOR pathway. And you can add other ones in there, but I think these are like the big giant ones to talk about, right? Obviously, sirtuins fail over time. We can bolster them. It helps. AMP kinase doesn't work as well over time, which is why the caloric restriction diet sort of helps because it turns that back on. Then the mTOR pathway is a whole nother category because there's a war raging about, of course, rapamycin and all of that. That's a fun topic all by itself. But let's see. So that's pathways. That's 3. 4 is something that I call quality control. And this is about checking to make sure that there's no damage and then the ability to fix the damage. So in this category, I have DNA repair mechanisms, protein repair mechanisms, and then the process of autophagy, right? So I always picture this as a factory model. And so this is like the checker system, right? These are the old ladies sitting there making sure your widgets are working. How do we fix the widgets, et cetera, et cetera, et cetera. Anyway, so that's category 4. 5 is your security system. And this has to do with your immune system. And of course, your cells in your immune system are very useful when you're young. And as you get older, they turn on you. And that, of course, leads to chronic inflammation. It also leads to more leukemias and lymphomas, as the cells are supposed to help you sort of become cancerous. And it also leads to more infections over time. So that is basically 5. 6 is what I call individual cell requirements. Because generally speaking, I pretend that all cells are the same, but of course we know that they are not. So in this category, we talk about the idea that some things are niche dependent. Some cells travel, some cells don't. A red cell has a different requirement than a liver cell or a brain cell. And in this category, I put senescent cells and then stem cells, right? Because we want to get rid of some, we want to keep others. And then the last category is what I call waste management. You probably realize that this is all the factory model to help people sort of keep track of it. So in my factory model, if you don't take out the garbage, your cell does not do very well. So in this category, it's mostly glucose problems. Of course, glycosylation is a huge issue around your body, which is why glucose or why diabetes is a big problem. And then, of course, lipofuscin accumulation in long-lived cells over time. So that is where I figure you age in 7 categories. And of course, there's subcategories. And various people move things around, but I think in this organizational pattern, it's the easiest way to sort of think about how a cell works. It's a little factory and it follows the rules of a factory. So that's my organizational scheme.
Yes. So have you ever tried to map that to the kind of the 9 hallmarks? Because there was that 2013 paper where they,
sure. So people talk about, like, Aubrey de Grey, he likes the 9 hallmarks. And they fit into my categories. But 7 is easier than 9. Proteostasis is essentially just protein repair mechanisms, right? If your proteins work, that's proteostasis. If they don't work, you have to fix them. That's quality control. Yeah. So everything fits into a category. It's just easier this way.
Right. Yes.
At least I think it's easier this way.
Yeah, no, it is definitely understandable putting them together. And yeah, I don't know whether telomeres deserve their own separate category or not.
No, they don't, because that's DNA damage. Right. Anything that happens in the nucleus to your DNA, at least in my world, is a DNA alteration. So that belongs in DNA category.
Right.
And the other thing, when I started doing this, I went to bench research articles. I didn't read anyone's synopsis. I didn't read Aubrey de Grey. I didn't read any of that. I stuck to absolutely bench research. So I wasn't influenced by anyone's decisions. I didn't even know there were the 9 hallmarks of aging when I came up with all this stuff. I did this in a vacuum. I did this sitting here in my office that I'm sitting in right now, simply reading through papers, writing down ideas, and then moving them around to make sense of it all. After the fact, people's like, oh, well, didn't you read such and such? And the answer is no, I didn't want to read someone else's synopsis because I didn't want to be their follower. I wanted to figure this out on my own.
Yes. So there weren't many papers on aging, particularly, I guess, earlier. So it was like you would come up with a tenet, okay, like mitochondrial function.
Well, so I would pick up a paper and it would say liver cell failure. Okay, right? So some poor little scientist is sitting in a lab somewhere, right? God bless them, because I could not do this. And they're identifying little pieces and parts of their cell. And they're like, ah, yes, this cell failed because it couldn't make protein X. Well, why didn't it make protein X? Well, it used to, and now it can't. It makes bad proteins. And you go, well, why did that happen? Oh, the cell ran out of energy. Well, why did that happen? Mitochondria failed. Well, why did that happen? And you follow these little chains down rabbit holes, and somebody is sitting in a lab somewhere and has figured it out, but no one's connecting the dots, which is what I tried to do, right? And granted, I talk about these 7 tenets as if they're completely isolated, but we know that they're not. I mean, you could sort of mix them around, little arrows everywhere, giant overlapping Venn diagrams. But in order to sort of understand them and be able to think about them rationally, you sort of separate them, knowing that it's completely an artificial construct. But someone has sat in a lab somewhere and dissected each and every one of these reasons. And that's amazing, because I just mooched off of their insights.
Right. Yes.